Assalamualaikum and good morning. Today we are going to look at chapter 5, cellular respiration and fermentation. Learning outcomes 5.1 A and B. We eat because we need energy. Fuel for cellular respiration in living organisms is carbohydrates, fats and proteins. Fats are excellent energy source because they contain rich hydrogen. Transfer of hydrogen in redox reaction will release energy. Role of respiration. Respiration will release energy stored in glucose and other organic compounds. Respiration converts this energy to produce ATP. ATP is a rich energy compound which acts as universal energy carrier of the cell. ATP is a modified nucleotide compound consists of three parts. The first part is one molecule of adenine, which is a nitrogenous base, one molecule of ribose, which is a 5-carbon sugar or pentose, and three molecules of inorganic phosphate groups. Structure of ATP. ATP means adenosine triphosphate, which means it has three phosphate groups. If the molecule have two phosphate groups, we call it adenosine diphosphate. And if the molecule only have one phosphate group, we call it adenosine monophosphate. The importance of ATP. It releases energy when hydrolyzed into ADP and phosphate in organic. When we hydrolyze ATP, it will produce 30.6 kJ of energy. When ADP is hydrolyzed, it will also produce about 30.6 kJ of energy, while AMP only produces 13.8 kJ of energy. 5.1b outlines the complete oxidation of glucose, which involves glycolysis, Krebs cycle, and oxidative phosphorylation. There are four metabolic stages in aerobic respiration. The first one is glycolysis. The location is at the cytosol or cytoplasm. There are no oxygen required for this stage. The second stage is link reaction or pyruvate oxidation. The location is at the mitochondrial matrix. This stage needs oxygen. The third stage is Krebs cycle or citric acid cycle. The location is also at the mitochondrial matrix. This stage needs oxygen. And the last stage is oxidative phosphorylation. The location is at the inner mitochondrial membranes, or we call it cristae. This stage also needs oxygen. ATP can be synthesized in three ways. The first one is substrate level phosphorylation, where a phosphate group is transferred directly from a phosphorylated compound to ADP to form ATP. The second one is oxidative phosphorylation, where energy released during transfer of electron used to bind inorganic phosphate to ADP to form ATP. And the last way is through photophosphorylation. ATP can be synthesized through substrate level phosphorylation, where a phosphate group from a high energy compound is transferred directly to ADP and it becomes ATP. The second one is oxidative phosphorylation. When hydrogen or electrons are removed from substrate, they are accepted by electron carrier. Example of electron carrier is NAD plus and FAD. These electrons are transferred to electron carrier in electron transport chain or ETC until it reaches oxygen, which acts as the final electron acceptor. Each step releases a small amount of energy, which is used to combine inorganic phosphate to ADP and to form ATP.